all the way from the University of Pittsburgh, Professor Robert Nicholls. Rob is a phenomenal researcher in the field of Prader-Willi syndrome. He is an expert in so many things. He was instrumental in discovering imprinting in general, but more specifically UPD and Prader-Willi syndrome. And I believe his current research is now looking at activating Prader-Willi syndrome genes. So it's both a personal and professional pleasure to introduce Rob Nicholls today and hear an update of his science and the potential to transform life with Prader-Willi syndrome.
Thank you so much. Are my slides there or?
Yes, they're ready to go, Rob. Can you see them on screen?
No.
Let me stop
Put
sharing.
the master up at the moment, I think.
Okay, let me progress the slide.
There we go. Thank you. It's gone.
I clicked one too many times. Apologies, Rob. Here it comes. And you have control to proceed.
Wonderful. Thank you. It's a pleasure to talk to everyone today, and I look forward to meeting many, if not everyone, tomorrow at the symposium. On this slide, I'm actually showing a picture of our children's hospital where my research lab is based, and the University of Pittsburgh. I'll go on. It looks like my slides are not advancing, so could we go to the next slide, please? Thank you. So as a quick summary of my talk, this is essentially also a quick summary of my career. I'm going to start with a little background about myself, what my relationship to Australia and Prader-Willi syndrome is, and then go on and talk about Prader-Willi syndrome as an imprinted disorder, which is where I began my research, and then go through some snapshots of different aspects of research that my laboratory's been doing, in particular, the use of animal models, what we can learn about Prader-Willi syndrome in human from the study of model systems, including cell models, which provide simple models for genetic manipulation. And since Prader-Willi syndrome is a imprinted domain of 10 highly conserved genes, it's not a simple genetic disorder. It's not one gene like many disorders. And so this adds complexity to both the understanding of the pathophysiological basis of Prader-Willi syndrome, and then also really what many of us are then searching for, which is how do we use genetic-based therapies or other therapeutic approaches to help improve the quality of life and the features of Prader-Willi syndrome for the individuals and the families. Next, please. So as the background, I'm actually from Australia. I grew up here. I was born in Ballarat, and some of you may know some of those little towns I mentioned. Those are all towns I lived in growing up, finishing off at high school. When we lived in Inverloch, I was at Wonthaggi High School. And so then I did my undergraduate degree, my Bachelor of Science Honours at the University of Melbourne, and majored in biochemistry. So I was in Australia till I was 22, and then left and went overseas to the University of Oxford, where I did my Doctor of Philosophy studies. I was based at Magdalen College in Oxford. If anyone's visited, it's a beautiful place. And my mentor for my PhD was Professor Doug Higgs. Just a phenomenal place to train. And after that, I moved on to the United States, and I did a postdoctoral research at Harvard or Children's Hospital Boston with the late Sam Latt. And it was when I started there in January of 1987 that I started working on Prader-Willi syndrome. At that point, very little was known. There was no genes mapped to the region. We knew the chromosome 15 deletion in a subset of patients, and that was essentially it. There was only a few clinicians, a few cytogeneticists that had even heard of Prader-Willi syndrome. Subsequently, after I finished in Boston, I've had faculty appointments starting at the University of Florida in Gainesville, and then Case Western Reserve in Cleveland. Spent 5 years at the University of Pennsylvania in Philadelphia, and then moved west in the state to Pittsburgh, where we've been since 2005. So that's a little background about who I am. And next slide, please. So when in Boston, I developed genetic techniques to look at the inheritance of Prader-Willi syndrome. And on the left, I'm just showing a single family. It's a DNA analysis. And you see a pattern of 3 different bands. The mother, there's 2 bands. The dad has 1 double intensity band at top. And the child with Prader-Willi syndrome, who had a chromosome deletion that was observed under the microscope, only shared the 1 band with the mom. And so the missing DNA is from the father. So that showed, and many families showed, that the deletion in Prader-Willi syndrome was always on the paternally derived chromosome. But we had a large number of families, about 1/3, that did not have a chromosome deletion under the microscope. And in applying the genetic techniques to these families, and one example is shown here in the middle, I discovered uniparental disomy. And you can see this. In this case, the band pattern is there are 3 bands. There's the bottom band shared with a sister, the mom, and the child with Prader-Willi. The father has a much larger band, and the second band in the child is the same as the mom. And so you see 2 copies, both derived from maternal inheritance. And so this was uniparental disomy, the first evidence actually in human disease for genomic imprinting. And so what we see on the right is, in the chromosome pictures, is on the left is the deletion on the paternally derived, the blue chromosome. And this occurs in about 2/3 to 3/4 of individuals with Prader-Willi syndrome. And then on the right is the 2 maternal copies of chromosome 15 in uniparental disomy, anywhere from 20 to even 40% in some countries now. And so it's the loss of paternal gene expression, the loss of this imprinted gene expression from this paternal chromosome, and it's missing here with 2 maternal chromosomes, that leads to the clinical features of Prader-Willi. Next slide, please. And so imprinting relates to a differential regulation of genes that depends on the inheritance from either the mom or the dad. And for Prader-Willi syndrome, the critical functional genes are inherited from the father. And this process you may hear referred to as epigenetics, which is a DNA regulation that alters expression of genes. Next slide, please. And just to show an analogy of what geneticists mean when they're talking about genomic imprinting and epigenetics, if you consider the pond, the DNA, the inheritance, the epigenetics is the covering, so the reeds and the lilies, and that's what's then regulating life in the pond, or the DNA regulation. So that's just an analogy to try to help understand this complex phenomenon. Next slide, please. And so uniparental disomy arises and is associated with an advanced maternal age. So this is the same mechanism as for trisomy 21 or Down syndrome, where you have a chromosome error during the development of the egg in the mom, which leads to a fertilized zygote, a fertilized egg, with 1 paternal copy and 2 maternal. Now, in trisomy 21, there'd be 3 copies of the chromosome, and that's compatible with live birth and a child with Down syndrome. For chromosome 15, 3 copies is lethal. So an embryo, a cell, can't survive with 3 copies. And just by chance, 1 copy is lost. And if it's the paternal copy that's lost, then you get left with 2 maternal copies. And so some societies in countries where women are having children at a much later age, then there is an increase in frequency of this mechanism. But Prader-Willi syndrome, the mechanisms, the uniprenar disomy, the deletion, and I won't have time to go into today, but the imprinting defects, they occur randomly in the population and in all populations of the world at the same level. Next, please. Okay, so I'm going to transition now to animal models, and the key point here is what can they tell us about human? Prader-Willi syndrome involves neurobehavioral features. It's difficult to study the brain to determine what's going on, and imprinting is a process that occurs in germ cells in the very early pre-implantation embryo, and again, it's difficult to study those stages in human. So by use of animal models, we can use a physiological organism that helps give insight into Prader-Willi syndrome in the human. And with that imprinted domain of 10 genes, 10 highly conserved genes, the same genes are found in most mammals. And so we can use animal models. Next, please. And so this is the first animal model that my laboratory studied. It's a mouse model of Prader-Willi syndrome, and it's a deletion mouse model. So that all the same genes that are deleted in human with Prader-Willi are deleted in this mice. And the Prader-Willi mouse is this little one on the left here that has dark fur next to its wild type or normal litter mate. And this is about day 5 of age. So the little Prader-Willi mouse is about half the size. The coat color is just a linked coat color gene, and it's not part of the genetic syndrome. And if we look at these mice, at the bottom left is a growth chart, and you see the wild type on top, and they're just before birth at embryonic day 18 and the postnatal day 1. They're a little larger than the Prader-Willi mice, and by day 5, they're double the size, as you see in the photo. The Prader-Willi mice grow a little, so they have milk in their bellies, but they're not taking as much milk, and they're not growing very much. So they're weaker, they've got less muscle, and they do have difficulty feeding. What's important with the mouse model is the survival. This is the middle chart is showing the numbers after birth of survival. And at birth, there's equal numbers. The orange is the wild type and the blue is the Prader-Willi. But by day 4, we've had some losses of the Prader-Willi mouse pups. But by day 7 to 8, there's no more survivors. And this is true of this deletion model and a couple of other complete genetic models, the imprinting defect model and a uniprenar disomy mouse model. And in our deletion mice, we found the basis, the reason that the animals don't survive is based on the blood sugar. At birth, the blood sugar is normal, and at postnatal day 1, the Prader-Willi levels are shown in blue. So by day 2, the blood sugar is already fallen low, and by day 5, it's exceedingly low. This is not compatible with brain function, and the animals are not surviving. Interestingly, after we discovered this low blood sugar or hypoglycemia in mice with Prader-Willi, other clinical workers went back and looked at charts and found that hypoglycemia episodes are not that uncommon in infants with Prader-Willi syndrome. And clinical colleagues tell me that they see episodes of hypoglycemia in adults with Prader-Willi. And in a few slides, I'll show a pig model that we also see very low blood sugar or hypoglycemia. So this may be an important part of the clinical features of neurodevelopment in Prader-Willi that we still do not have a good clinical understanding. And I think it's going to be important to look into this further. Next slide, please. Back one. There we go. So we've done a lot of studies on the metabolic and endocrine and nature of the mice. And the surprise was that we didn't find initially anything obvious about abnormalities in the hypothalamus or the brain. But what we found was a endocrine and metabolic defect. So I mentioned the low blood glucose. We also found that the Prader-Willi mice were deficient in insulin levels and in glucagon levels. And these are two hormones that are involved in regulation of blood sugar, and blood sugar also is important for regulating eating and hyperphagia. What I'm showing on this slide is a defect in the pancreas in the beta cells that produce insulin. And what we did was we used a fluorescent protein that marks the synthesis and secretion of the hormone insulin. And on the left, in the control, the cells that are green and a little yellow, this is newly synthesized insulin. This fluorescent protein is really quite magic because it changes from green to red over 24 hours. And when we looked at the litter mates of the controls in the Prader-Willi mice, their beta cells are all red. So that shows that the insulin that's been synthesized is aging and it's not being released. So there's a defect in release of insulin. It's important to recognize that from Prader-Willi mouse models, they've taught us a lot, and a lot of different workers in Prader-Willi syndrome field have studied individual gene knockouts in mice. They typically have a very mild phenotype, and but none of the genetically complete models survive more than the 1 to 5 days. And so we can't use the mouse to model hyperphagia and other behavioral aspects of Prader-Willi. Next, please. I've just put one slide in here to show we recently published work in which we established a beta cell model of Prader-Willi syndrome. So we used a process called genome editing to generate deletion of all the Prader-Willi genes, and then looked at the beta cells in culture. And what we're showing here is a chart that looks at insulin secretion, comparing control to Prader-Willi, and you see that both at low glucose and then at high glucose, which is glucose-induced insulin secretion, like after having a meal, that the Prader-Willi levels of insulin secretion are lower than the control. And we saw exactly the same thing in our Prader-Willi mice. And so these cell models are mimicking what's going on in vivo. And we were then able to use the cell models to look at every RNA and every protein expressed in the beta cell. And that allowed us to identify defects in a specific class of proteins that are actually involved in hormone secretion. And so going on now to try to understand why those proteins are defective in their production in Prader-Willi, and if we might be able to utilize that for a therapeutic development, perhaps of a small molecule. So going on to the next part of the talk, we developed another animal model. So again, the mice with Prader-Willi weren't surviving, so we needed a larger animal model in which brain development was more like that in human. And this slide just shows an example. On the left, you see brains from rat, rhesus monkey, which is a non-human primate that's often used in research, pig and human. And on the right are sections through the brain. And what you see is that the rodent brain is very smooth, whereas the human brain has large folds in the cerebella. And this is the increased degree of folding is associated with increased neural complexity and neurobehavior. And what you actually see from this slide is that the pig brain is both larger in size than the rhesus monkey and has a more folding pattern, more like human than does the rhesus monkey. So while some people suggested that a non-human primate's the optimal model, this analysis would actually suggest the pig should be a better model for neurobehavioral studies than a lower non-human primate. Pigs are very smart animals. And so next slide, please. And if you look at in the table, I've listed all the clinical features of Prader-Willi syndrome, and all these clinical features have been studied in pigs or can be studied in pigs. Pigs are a good model for metabolic syndrome, which involves obesity, diabetes, and atherosclerosis. They're the best animal model there is. So pigs should be a good model for Prader-Willi. Next, please. And so we use genome editing, in which we can go in and specifically modify the genetic process that involves the imprinting process in Prader-Willi. Next, please. And this just shows an example. It's a DNA gel, and the bands at the very top are the normal bands. And I've just shown an analogy of a race car. We can tell that as a geneticist, whereas all these different sized bands at the bottom are the genome edited deletions. And that's basically a broken gene, like this race car that's smashed up. So that this methodology allows us to generate the model of Prader-Willi syndrome. Next slide, please. And then we're able to, using the same techniques actually that were used to generate the COVID vaccines, we use that technique by injecting the RNA that allows the genome editing into the fertilized zygote, transfer those embryos to a surrogate female, and then obtain a litter of pigs. Next slide, please. And by doing this, we actually had 2 successful litters. The one on the right is the critical one. It actually represents 1.5 Prader-Willi pigs. The full Prader-Willi pig is 120-2. You see his muscles are weak. He had a full genetic mutation in all his cells. The half Prader-Willi pig, the little guy on the right, was because he was a mosaic. Half his cells had Prader-Willi and half were normal. They had the full Prader-Willi pig had growth retardation and developed episodes of severe hypoglycemia, but we were able to treat with a glucose solution. Next, please. And those pigs grew up, and the partial PWS is on the left. A full PWS is on the right. What's really distinct, you can see his severe scoliosis, which is a important part of, for many people with Prader-Willi syndrome. And so the pig should be a good model to study this aspect. Next, please. We did this work. We generated the pigs in collaboration with a group in Missouri. And this is a weight chart showing the growth in weight. The control, well, he's not really controlled. The half Prader-Willi pig's in red, and the blue is the full Prader-Willi pig. So we don't have controls on here. In Missouri, they kept the pigs on a very strict maintenance diet. When we transferred them to Pittsburgh on the right, our vets said, oh no, we can't control the diet. We've got to let them have ad lib feeding. Well, you see what happens as soon as we let them have free access to food. Their weights just really ballooned and took off. And our vets said, hey, we got to get this under control. The pigs are getting too big. They're dangerous for the workers. So we put them on a maintenance diet, and they leveled right back out. But this suggests there's probably hyperphagia, but we weren't able to do a controlled study with such a small number of pigs, and we didn't have controls of the same age. Next, please. And so this shows that same list of clinical features, and the arrows represent the clinical features we have observed to date in the pig model. So the failure to thrive, the muscle hypertonia, episodes of hypoglycemia, probably hyperphagia, but we need to study that in the future. We have not yet looked at being able to look at body composition. We do observe, we've just initiated a study looking at the hypogonadal tropism, and there are defects in production and function of sperm. A full Prader-Willi pig had severe scoliosis. They also had problems with temperature regulation and anesthesia complications. We have to do anesthesia each year to do a vet checkup, and we had to be very careful bringing them back from anesthesia because they developed problems with temperature and hypoglycemia. Next, please. So the pig model could be a great model to study neurobehavior, but pigs are very expensive. That is a major problem at a major medical center. It's best if you have an associated farm to keep the costs down. Many pigs are still very large. So we're still considering other animal models. FPWR in the US, I understand, have helped fund the development of a rat Prader-Willi model. I haven't seen that published yet. And I'm considering other models. We've actually done some cell studies to consider the potential of a cat model, a much smaller laboratory model that will be much easier to work with, but still able to do detailed behavioral studies. But we need funding for that. And there's even consideration of other potential models that can give insight into different aspects of hyperphagia, such as animals that hibernate. Next, please. And the last section of the talk now, I popped this in. It's actually a electronic art from my daughter. It's showing fireflies at night. Everybody, I think, is familiar with fireflies. The light that's produced is from a protein called luciferase, and we use that for our genetic studies. And in the next couple of slides, I'll show some data that is using luciferase expression, this light production. Next. And so this is what we're proposed to do, is rather than trying to activate the silent copy of the Prader-Willi genes, which others are doing, and we're going to hear about today, we've taken an approach where using synthetic biology, where we're actually able to now encode up to 8 of the 10 highly conserved Prader-Willi genes in a single AAV or adeno-associated virus vector. And we've been able to do this by miniaturizing components. And the little red bars are a miniaturized promoter we developed to help fit in the synthetic miniaturized Prader-Willi genes. Next, please. This just shows an example. On the left, you see a set of DNA constructs, and the yellow bars represent motifs for a specific transcription factor. And the more bars we have, the higher the level of luciferase production they produce. And you see that by the green bars. And so this has enabled us to really decrease the size of some of the components for the AAV vector. Next, please. And just to show some examples, these are these small, very small miniaturized promoters driving green fluorescent protein, and we also have luciferase, but I'm not showing that data. And so you just see all the green cells lit up, and these are beta cells, and we've also done it for alpha cells, fibroblasts, myoblasts, which are muscle cells, and neurons. Next, please. And then with my collaborator, Tina Zhang and George Giddies, we used an AAV that encodes GFP and luciferase and injected them into mice. And on the left panel, on the right, we see injection into the tail vein, which then targets the liver. And so by dissecting out the different internal organs, we can prove that that's the liver that we see expression. But the virus can be injected directly into the pancreatic duct. And when we do that, we see the luciferase, the light production, and that when we dissect out organs is in the pancreas. So we can target this gene vector directly to the pancreas. And so that's what we're doing. I have an NIH funding to look at correction of the beta cell phenotypes, the insulin secretion defect, and the other protein defects were found in the beta cell model. And then we're using it to correct the survival and the endocrine defects we see in the mouse model of Prader-Willi. Next, please. And this is just showing that we have the ability to inject in mice where you have litters of 6 to 8 or even up to 10 pups, so we can inject a subset of the developing mice and then observe after birth the clinical phenotypes. We don't need to worry about the fine details of this slide. Next, please. So I'll finish up here. I just wanted to try to give a snapshot. Human genetics, while my first decade or 15 years or so was studying human genetics, we've transitioned into the other types of work I mentioned. David Gogler, who's going to talk to us, has developed newborn screening for Prader-Willi and other epigenetic disorders, which is going to be tremendous for the population. animal models are an important part of our work, and we're continuing to work with those we and others have developed, and new models we're trying to develop. We're going to continue with cell models. There are still various cell types that, for example, neurons that secrete oxytocin, which seems to be defective in Prader-Willi syndrome, that needs to be better studied. And then based on the increased genetic knowledge and the approaches such as the AAV approach we're taking, and the gene, the silent gene activation that others are taking, we hope to be able to develop therapies. And then from some of the work, like our beta cell endocrine work, we also hope to be able to develop small molecules that may be applicable for treatment. And so I've been working on Prader-Willi syndrome for 37 years. So my goal is to do at least another 13 years and reach 50 years. And hopefully by that point, we'll have really good treatments that really allow people with Prader-Willi to reach their full potential and not deal with the clinical difficulties they have. Thank you, and I'll be happy later to take questions.